But you see, the point is you see faster growth rates at higher temperatures, and that rate of increase is the slope of that is usually quite predictable. We see it among species, we see it within species for larval duration, and we see it among different groups. So to say that temperature causes rates to accelerate identically is still not actually super useful for understanding community patterns, because it just says everything's going to speed up and be the same. So it becomes interesting when this part isn't actually true. So that's what I'm going to talk about. So we talk about asymmetries and thermal dependence. And this is providing quite a way forward in looking at complex ecological systems and how they change with temperature in terms of a few key thermal asymmetries. So this one that I'm interested in has to do with the difference between the response of autotrophs and the response of heterotrophs to warming. So basically heterotrophic respiration-limited metabolism is about twice as sensitive as photosynthesis. So there's a theoretical argument for this. There's also an empirical argument for it. Rose and Karen in 2007 published a paper where they updated the Epley curve and found exactly the same curve, and they also thought they would look at heterotrophs, and they found that the exponential increase in maximum growth rates for heterotrophs is steeper and about twice as steep. And they published a whole paper and never mentioned anything about metabolic theory, but someone else wrote a letter in response and showed that the slopes, or the parameters of these curves, match this prediction exactly. So we have a lot of data to support that, at least for single-celled organisms. All right, so as a community ecologist, then in graduate school, this was super exciting because my supervisor had been very skeptical about looking at temperature effects, and my favorite quote is, he said in my first year, he said, well, what are you going to do, just warm things up? We'll just get faster. And then this gave me a way to say, well, actually, no. This looks like herbivores and plants are going to respond differently to temperature, and that there's potentially a temperature dependence of herbivory. So I tested that, having the prediction that the herbivore-plant interaction strength gets stronger with warming. I did that in an experiment using seaweed, or amphipods that graze seaweed. And I love this picture. It just shows, this is sort of a storage tank where these guys were. It just shows how effective they can be. These are their little tubes. Here's one or two of them. And you can see the grazing halos, and there's no algae around in their little landscape. But this is the water line where they couldn't persist, and there's thick beds of algae. So these guys can be really strong grazers in a short amount of time. And I did a lab experiment for 2 weeks with the prediction that interaction strength between herbivores and plants gets stronger and more negative with warming. And that's exactly what I found. So these are sublethal temperatures. The temperatures that I tested were within the range of what they would experience in a week at that time of year, and the interaction strength got stronger. So this doesn't get us to abundance, sort of this bigger picture understanding, but it's the first step in that direction. So the question, remember, is do physiological and now demographic responses interact to determine the effect of temperature on abundance? So if we're interested in abundance, of course, we need to remember that abundance is a dynamical outcome. We can't just go from physiology to abundance. It doesn't really work that way. Temperatures effects depend on the dynamics underlying abundance. So to think about this is this great schematic figure that Dave Vasseur actually sketched out, and I stole it because it's so useful. Physiological response scale, temperature affects body temperature of an herbivore and a plant. Those then, body temperatures might affect vital rates, demographic vital rates, and this is sort of the scale at which interaction strength is relevant, right? But of course, vital rates affect abundance, and if these guys are interacting, their interactions are also going to affect abundance. And then that we might measure as population size. And then if you run it long enough and you have a stable system, et cetera, you might have an equilibrium abundance that's going to reflect everything that's happened along the way. So the next experiment I did was to look at population size. And the question here was, does the heterotroph-autotroph asymmetry control population abundance over a temperature gradient? So using planktonic systems from the estuary where we worked, so a bunch of mesocosms in different temperatures. The design was a nutrient addition and a nutrient control treatment. I'll just talk about the nutrient addition results, but you'll see the data for the controls, and we can talk about that later. The results, so the hypothesis was that productivity and respiration would change with temperature. This next say productivity. This is primary productivity, milligrams carbon per chlorophyll per hour. It did increase with temperature when the nutrients were not limiting. So the black symbols are those nutrient addition treatments. So as we predicted. So, but this corresponded with a decline in phytoplankton abundance in milligrams carbon. So the initial conditions in this figure are the black line, that's what I added. And after 8 days, we had a decline in phytoplankton abundance with much higher abundances in the colder temperatures. And this is despite increased productivity, right? So the hypothesis was like research would be driving this, and we did see, is here's zooplankton density, we saw a disproportionate increase in zooplankton abundance over the temperature gradient, sufficient to drive down the phytoplankton abundance despite their increased productivity. And we saw the same results, right? So primary productivity increased, but abundances declined due to grazing. And we saw the same trend in bacteria, the other heterotrophic compartment in this food web. Okay, so we've shown, so we're kind of building up. So interaction strength, population size. This population size result is really for here after 8 days. We didn't allow enough time really for dynamics. So the next question is, well, what would we expect if we had a longer term system? So to do this, we turned to theory, and I collaborated with Ben Gilbert, who was a postdoc in Santa Barbara at the time I was there. And we used a basic consumer resource model, and we tested a bunch of different ways you could write this model. These are the general terms where plant growth increases with plant population growth and it losses to consumers, and then herbivore growth increases with its consumption at transfer efficiency and losses to mortality. And we just gave the plant term, the autotroph temperature dependence, and we gave the consumer terms, the heterotroph temperature dependence. And we ran out these models to see how abundance would be expected to change with temperature. So I'll show you the results on figures that look like this, with log of abundance on the y-axis and then temperature on the x-axis. And for a system without a consumer, we might expect phytoplankton abundance to decline with warming. And we can predict the slope of that decline as that activation energy, that temperature dependence of autotrophy. That's the model sort of output. So not only do we predict the direction, but the rate of decline with warming of phytoplankton abundance. So our initial concern was maybe, well, systems with different dynamics might each give you different results when they warm, and therefore you'd have predictable effects of temperature on organismal physiology, but not predictable effects once they go through dynamics. You might get any possible outcome. So we were excited when we didn't see that. So these are the 4 models that gave us stable solutions of the 9 that we tried, or 9 that we tested. We only saw 2 types of outcome. One type had stable plant abundance and declining herbivore abundance with temperature. Another type saw parallel declines, or predicted parallel declines in plant and herbivore abundance with warming. And not just declines, but we predicted the slopes of these declines. The difference between these 2 outcomes has to do with whether mortality is density dependent for the herbivores. The other terms are interchangeable in terms of the outcome you get. The slopes for any of these declines is the difference in activation energy. The difference in the temperature dependence of heterotrophs and autotrophs gives you this rate of decline. So even if they're not what you predict from metabolic theory, if you measured them for your system, that should give you your prediction for how abundance would change. And the reason for that is algebra, basically. Here's the temperature dependence term, and here's all the biology. So you don't need to know the growth rate, the carrying capacity, the attack rate, etc. You don't need that. All you need to know is which model describes your system, and that gives you a prediction for how it will change with temperature. So it's quite simple. This one is the one people usually use to analyze mesocosm experiments. The assumptions fit the assumptions you would make for a mesocosm. So we used it to test against my data from the previous experiment. This is the prediction for the long-term dynamics, and I'm showing you them here run out through time. So what we have in this figure is a cool and a warm, so herbivore abundance is in brown, and plant abundance is in green. And you can contrast a cool herbivore assemblage with a warm one by moving from the solid to the dashed lines. So the effect of temperature is the contrast between these 2 lines and these 2 lines. So you can see that this no change in trajectory through time for phytoplankton here looks, the average of those 2 curves is not different, whereas it is for herbivores. And I'm showing you this to show you where my experiment falls. So the experiment only ran for 8 days. We know what we started it with. We know when it ended. So we parameterized this consumer resource model with our temperature control, so the unheated, and then we asked how well temperature could predict the abundances at other temperatures. And we gave it these activation energies derived from, or borrowed from metabolic theory. So these are the results on the same axis. We have temperature here, zooplankton density, phytoplankton density. The open circles are our replicates from our mesocosms, and the filled symbols are the model predictions, except for the low temperature. So the model did a good job of predicting the effect of temperature on these final abundances for our experiment, given that we had good reason to think that was the right model to use. So this was really encouraging, and it also helped us combat a few criticisms about the relevance of mesocosms, the permanence of their results in time, and it shows what, of course, if you think mathematically about dynamics is obvious, but to empirical experiment people, it's less obvious, that you can get short-term results that are very different than long-term results, but are underlain by the same dynamics. So this gave us encouragement to move forward, and by move forward, we then continued this joint use of theory and experiments in a working group, and I would love to tell you about all the cool stuff we did, but I won't. I'll just tell you, if you're interested, to go look for them. One is a temperature-dependent consumer resource modeling framework that just came out using sort of Kevin McCann's bioenergetic approach to food webs, but doing it to ask this question about temperature dependence. We also did some work on looking at fitness across latitude, so if you still think that tropical species are more vulnerable to warming than temperate species, please read this paper and several others that have shown that that's not true. And species interactions in many communities with warming. And the thing that my lab does, that I'll touch on in the last few minutes, is test these kinds of expectations. So we really work, my students and postdocs work on experiments to take something like this framework that I just walked you through and say, okay, we can understand this reasonably well. What happens when we scale it out even further and put this set of dynamics in an ecosystem with and without predators? Or we run it for even longer, or we add competitors. So one example of this that I'll show you is ask the question, does food chain length alter the effects of temperature on simple ecosystems? So I'm introducing also this sort of energetic ecosystem response. This project was done by Jessica Garska, a visiting PhD student a couple of years ago. And basically the framework for this is that I've just told you how temperature affects the outcome of trophic interactions, and we know that from many other experiments. We also know that trophic interactions affect ecosystem fluxes. Great work by Daniel Schindler and others show that when you have a top predator, you can have changes in the total carbon flux in the system. And we know, we have very good evidence that there's a clear effect of temperature on ecosystem fluxes across ecosystems, such that when you warm, ecosystem respiration goes up and the slope is predictable by, it's the metabolic theory prediction. But at the community scale that we want to understand, if we're monitoring or tracking it in the context of climate change or whatever, we might be concerned about how these things interact, right? Sure, across ecosystem temperature has this nice signal, but what's going to happen in my system where I have a predator or whatever? So our questions here, does temperature modify this pathway, or do trophic interactions modify this effect of temperature on ecosystems? And we tested this using, because we're motivated by this sort of theoretical prediction of slopes, we need many temperatures to fit a functional response to temperature. Two won't do it. So we have many temperatures, and we have a one trophic level system where we have microzoa, well, we're calling it a one trophic level system in phytoplankton. We had another set of replicates, or another set of temperatures where we had grazers, and a three trophic level system. So we have this cross between food chain length and temperature. And we added nutrients at the beginning to create some sort of reasonable nutrient ratio. We ran it for 8 weeks. So the results are that fluxes scaled positively with temperature independent of food chain length. So food chain length, these are what the results look like. I'll just show you all of them at the same time. So here's net primary productivity at our ecosystem scale, net ecosystem respiration. The slopes of these, the different symbols are the different trophic treatments, 1, 2, or 3. You can see that they, this is for week, 1 week by the way, maybe it's week 8, I must have. The net productivity and respiration rates increase with temperature. The absolute respiration or productivity depends on trophic structure, but the effective temperature does not. And that's true for both. And the red line is the predicted slope based on metabolic theory. So we do see a difference in the slope, but not due to trophic level. So these are the fluxes. I was previously talking about dynamics, right? So we're wanting to know how these go together. The zooplankton trajectory is qualitatively similar to our prediction. We see a decline in zooplankton abundance with warming, as we predicted and saw for a true 2 trophic level system. But the effects of predators, we see effects of predators on that temperature dependence of grazers, and the effect of predators is much stronger at cold temperatures and basically disappears at high temperatures. So there is an interaction between food chain length and temperature on the abundance of grazers. And there's even some other details. So the community was changing a lot with temperature. Predators I just said that. The other thing is that we see a community shift. Daphnia dominated all mesocosms at cold temperatures. Daphnia fell out, and the systems were dominated by copepods at warm temperatures, and the temperature at which that switch happened was lower with predators. So we see this interaction not only in abundance, but in composition of the grazers. And we also observed smaller body sizes with warming, and we've seen that the size shift in individuals within Daphnia is both demographic and evolutionary. Despite all of this cool stuff happening in our populations and communities in this system, we did not see that these trophic interactions or temperature modified this overall framework that temperature constrains the fluxes at the ecosystem level. So all those dynamics are happening within this broader constraint. So I could tell you much more about those, but I won't, so I'll wrap up this time. So the science summary, I talked about a lot of different things. So the main take-home sort of scientific findings are that local scale species richness has increased recently, recent benthic breeding systems, and it's likely reflecting invasions and range expansions. Still working on the details. Sublethal warming can strengthen herbivory, affecting herbivore abundance and herbivore and plant dynamics and abundance. And then finally, effects of temperature on fluxes in our ecosystem experiments were resilient to the effects of temperature on species interactions. So I'm going to skip that for now because I'll say it here in my sort of soapbox summary, my opinions that I started with and we'll end with are that detecting and attributing ecological change requires evidence-based expectations, right? So the science that we do in the different forms can fit into this broader framework, and it gives us a way to talk to each other, even if we're using different methods at different scales. So let's use theory, experiments, and observations to derive expectations and to articulate them that way, and that's something that's inclusive, something we can all do in this context of global change. So that's it. I have many collaborators to thank: biodiversity analyses, the climate impacts group, and the people who gave us data for the diversity study.